Hello and welcome to National 5 Biology, Unit 2, KA1, Producing New Cells. So we're still on Unit 2, Multicellular Organisms, and this is Topic 1. Once again, here's the SQA course specification, or mandatory knowledge section for this topic. So this is all the bits of knowledge and content that you could be tested on in the test or final exam for this level. As you can see, this is a slightly larger topic, so feel free to pause the video at any point or go back and rewatch sections if you need to. I've broken it down into two parts, so as we go through, we can go through the theory and we'll do some questions after each part to make sure you know you're ready to move on. Now, our first bigger topic within this key area is mitosis. So our learning intention today is to learn about producing new cells, and hopefully by the end of this lesson you'll be able to describe the sequence of events in mitosis, explain the terms chromatid, equator and spindle fibre, and explain the importance of mitosis. So firstly, this is just a reminder of what you should know already about DNA and chromosomes from way back in Unit 1, Key Area 3. So if we imagine that this is inside the nucleus, here is the chromosome with the characteristic X shape you're probably used to seeing by now, and this is in fact made up of DNA, which has been coiled and condensed to form this chromosome structure. Now, this chromosome has already been replicated or duplicated, and each side of this X can be referred to as a chromatid, but we'll come back to this in more detail soon. So before we look at chromosomes in a bit more detail, we need to know what a diploid chromosome complement is. So most cells are what we call diploid. Diploid means that the cell's nucleus contains two matching sets of chromosomes, and this is really important. So here on the diagram on the right-hand side, you can see there are three matching sets of chromosomes here. Don't worry about haploid just yet. We'll come back to this in more detail and a bit more about diploid as well when we do Key Area 3 reproduction. So diploid cells have two matching sets of chromosomes inside their nucleus, and these need to be replicated during a process called mitosis, which is our focus in this lesson. So if we think about a cell that has, for example, 46 chromosomes like ours, when our bodies go to make new cells, they want those new cells to contain all of the same genetic information that was in the original cell. So in other words, we want each cell to still have the 46 chromosomes that contains all of the genetic information that codes for us. So every time a cell divides, which is what mitosis is, the parent cell splits and creates two daughter cells. And these two daughter cells need to have the exact same number of chromosomes in their nucleus as the original parent. So in this example here, there are two chromosomes and the daughter cells both have two chromosomes each as well. Now this is essential because we don't want any genetic information to be lost. If this original parent cell just split without making new copies of its genetic material or chromosomes, each of the daughter cells would only get one out of the two chromosomes, so half of the genetic information would be lost, which wouldn't be a good thing. If this happened each time our cells divided, we would lose half of our genetic information each time, and so we'd go from 46 to 23 chromosomes, and then even smaller than that, which again is not what the cells want. So at the start of mitosis, the original cell, the parent cell, duplicates or replicates its chromosomes, so it has copies, and then these can be split between the two daughter cells, so they both get the same genetic information and no information is lost. Now you might notice that the chromosomes in the parent cell and the chromosomes in the daughter cell in this diagram are different shapes. So let's explain that just now. So you need to know the difference between chromosomes and chromatids. So normally the chromosomes in cells look like the diagram on the left. Here we have two chromosomes. Now if these were split during cell division, each daughter cell would only get one, and then genetic information would be lost. So how does the cell stop this from happening? Well, right before mitosis takes place, the cell duplicates or replicates the chromosomes and makes these characteristic X-shaped chromosomes on the right-hand side that you'll have seen before. These are still both chromosomes, but they are now chromosomes made up of two identical chromatids. So a chromatid is half of the duplicated chromosome here. So this side is one chromatid, and this attached in the middle to its identical partner chromatid on this side. And this means that one half can go into one daughter cell and the other half can go into another daughter cell. And it means that both of them will end up with the same number of chromosomes still. So both daughter cells in this case would still have two chromosomes. So this is one chromatid, this is the other identical chromatid, and this is one chromosome. Again, this is one chromosome made up of two identical chromatids. These chromosomes only have one chromatid, so there's just one chromosome. So you'll see this in more action as we go through the stages of mitosis, but for now both of these diagrams show 2 chromosomes, but the chromosomes on the right have been duplicated, so they are now 2 identical chromatids joined together. So now we're going to go through the stages of mitosis. These are really important to remember. They could be an extended response question, or they could be made to put these in order or describe what stage comes next. So this is our original or parent cell. This bit in the middle, this bit that's shaded in here, is the nucleus, and it's filled with uncoiled chromosomes ready to begin mitosis or cell division. By the end of mitosis, there will be 2 identical daughter cells produced from this one with the exact same chromosome complement or number of chromosomes within the nucleus. So by the end of this, we should see 2 daughter cells which are identical. So in stage 1, within the nucleus, so this part here, each chromosome replicates or duplicates to form chromosomes made up of 2 identical chromatids that are joined together. So in this example, there are 4 chromosomes, each made up of 2 identical chromatids, but remember, depending on the type of cell, there could be different numbers of chromosomes each time. For example, in our cells, there would be 46, but to keep things simple, we're just going to stick for the 4 in the example just now. So we have 4 chromosomes, each made up of 2 identical chromatids. So in the nucleus, each chromosome replicates, and remember, this is so we don't lose any genetic information later on once the cell starts to split. In the next stage, you can see that the nucleus has now disappeared from my diagram. If I go back and forward here, you can see the dotted line of the nucleus here, and now it's disappeared. So the nuclear membrane has dissolved, and the chromosomes, or 4 chromosomes we talked about before, have now lined up along the middle of the cell. So this is not the middle of the nucleus, this is the middle of the cell, and this part of the cell is called the equator. The way to remember this is to think about a globe of the world. The equator is the part around the middle. So the chromosomes line up along the equator once the nucleus is dissolved. Now these dotted lines that are coming down from the top and the bottom here are called spindle fibres, and they attach to each chromosome or pair of chromatids. So the nuclear membrane dissolves, the chromosomes line up along the equator of the cell and are attached to spindle fibres. In stage 3, you can see that the spindle fibres have pulled the chromatids apart, and the chromosomes are moving towards opposite poles of the cell. So a very important part here is the difference between chromosomes and chromatids. An easy way to remember it is that you never say chromatids until this stage here. So only refer to them as chromatids when they're being pulled apart by the spindle fibres. Before or after this, just refer to them as chromosomes. So the spindle fibres pull the chromatids apart, and the chromosomes move towards opposite poles of the cell. The poles are like the poles on a globe as well, so just the top and bottom parts here. So now the chromatids have been pulled apart, and now the chromosomes have moved towards each side of the cell. We now have the correct number of chromosomes to create the 2 new daughter cells. So now that the chromosomes are normally contained in the nucleus, we know that normally. So although that dissolved in the last stage for it to happen, now 2 new nuclei are forming. So we're getting 2 new nucleuses being formed here. So the nuclear membranes reform, creating 2 nuclei, one for each of the new daughter cells, and the cytoplasm starts to divide. Eventually, the cytoplasm splits and 2 new daughter cells are formed. Both of these daughter cells have the exact same chromosome complement or number of chromosomes as the original parent cell, so 4 in this case. And then the process of mitosis starts again. So each of these cells will become a parent cell, and that will split into 2 new daughter cells through mitosis. So these 2 will split and make 4, and then that'll keep happening again and again. Now that was a lot of information, so feel free to replay those stages again if you still aren't sure of how to describe them before moving on. So one final reminder, both of these first 2 images are single chromosomes, but the second image is a chromosome that has been copied itself to create 2 identical chromatids that are joined together at the middle. The only time you call them chromatids is when they're being pulled apart by the spindle fibres, and then they go back to being chromosomes again, and then this process begins again. The last thing you have to know about mitosis is why it's important. There are 4 main reasons that mitosis is important. The first 3 are to do with mitosis providing a new supply of cells. So every time a new cell is made in the body of a multicellular organism, it'd be through mitosis. These new cells can be used for growth, repair of damaged tissues, or replacement of dead or damaged cells. Easiest way to try and remember this is GRR. So growth, repair, replacement are the three of the main reasons why mitosis is important. Another reason mitosis is so important is that it maintains the diploid chromosome complement of the cell. So in other words, no genetic information is lost each time a new cell is made. The number of chromosomes stays the same, but you need to work as it maintains, basically word that as it maintains the diploid chromosome complement. You might be able to explain that in slightly different words, but if you just remember it maintains a diploid chromosome complement, that's enough. So there are the 4 reasons that mitosis is important. So let's try some quick questions on what we've covered so far to check our knowledge before we move on. So pause the video here and try these questions either by saying the answer out loud or writing them down. And when you're ready, play the video and we'll go through the answers. Okay, so for the first question, it asks to rearrange the letters so the stages of mitosis below were in the correct sequence. This is a really, really common type of exam question. If we were to put these in the right order, E would be first, then C, because that's showing the chromosomes duplicating. Then it would be D, so chromosomes line up at the equator, nuclear membrane dissolves, spindle fibres become attached. Then it'd be A, the chromatids being pulled apart, chromosomes moving towards opposite poles of the cell. And then finally it would be B, so that's where the nuclear membranes reform 2 nuclei and the cytoplasm starts to divide. Name the structures which pull apart the chromatids towards opposite poles of the cell. That's spindle fibres. Name the name given to the area of the cell where the chromosomes line up. That's the equator. Give 3 reasons why mitosis is important. So you can give any of these. So maintain the diploid chromosome complement to provide new cells for growth, repair of damaged tissues, or replacement of dead or damaged cells. So remember, growth, repair, replacement, and maintaining diploid chromosome complement. Okay, so before we move on to stem cells, there's a really common exam question on this topic which relates to cell division calculations. If we think about it, every time a cell divides through mitosis, 2 daughter cells are produced. So each time a division happens, the number of cells doubles. These calculations either ask for the number of cells that are produced in a given time, or for the time it takes for a certain number of cells to be produced. So we're going to look at 2 examples of each of these styles of questions. The first 2 examples look at how you would calculate the number of cells if you were given the time. So in this example, it says, if a cell divides every 20 minutes, how many cells would be present after 1 hour? Now to work this out, I like to lay out my answers like this every time. So at the top, I write how long it takes for each cell to divide, or in other words, how long it takes for the number of cells to double each time. Then down the left, I write the times going up in number of increments I just wrote in. So in this example, it says the cell divides every 20 minutes. So starting at 0, I write 0, 20, 40 in 1 hour. I then stop there, and the question is now asking me how many cells there would be after 1 hour. So I stop at that point. Then on the right-hand side, I start with the 1 cell, and I double until I get to the end. So I double 1 is 2, 2 doubled is 4, 4 doubled is 8. And so how many cells would be present after 1 hour? That would be 8 cells. This is the easiest way to get the correct answer. Some people try and do shortcuts, but I've seen them trip up with these types of questions, especially if they get a bit harder. So this is the way I would always suggest laying it out. So let's try another example of this type. So in this example here, it asks if a cell divides every 30 minutes, how many cells would be present after 3 hours? So it's asking about how many cells again, but the total time has changed, and so is the time when the cells divide. But we still approach this in the same way. So at the top, I note that the cell divides every 30 minutes, and then I start to write in my times. So I always start at 0, and then I go up in 30-minute increments until I get to 3 hours, which is what it was asking about in the question. So 0 minutes, 30 minutes, 1 hour, 1 hour 30, 2 hours, 2 hours 30, and then 3 hours. Then on the right-hand side, I start with 1 cell and I keep doubling until I get to the bottom line. So 1 doubled is 2, 2 doubled is 4, 4 doubled is 8, then 16, 32, and finally across from 3 hours would be 64. So my final answer for this question is 64 cells. Okay, so now we're going to change the style of question a bit. This time the question asks if a cell divides every 10 minutes, how long would it take for there to be 64 cells? So this question is different. They want us to tell them the time and they've given us the number of cells. But actually this works in the exact same way as we did before, but we just do the right-hand side first this time. So I'm still remembering here that the cell divides every 10 minutes, and then down the right-hand side this time, I'm going to start with one cell and then keep doubling until I get to 64, which is what it asked about in the question. Then I go to the left-hand side and start writing out increments of 10 minutes until I get to the bottom, and then that'll tell me the time. So 0, 10, 20, 30, 40, 50, and 1 hour. So it takes 1 hour to make 64 cells. So 1 hour is my final answer. So one final question, which is the hardest one they might give. This question asks, if a cell divides every hour, how long would it take to get 256 cells from the 16 original cells? So this question is the same structure as the last one we did. However, this time we aren't starting from one cell like all the other examples. We're now starting with 16 cells. So the cell divides every hour. Starting with the cells first, we are beginning with 16 cells, not just one, and we're just doubling each time until we get to 256. So we're starting at 16 and we're aiming for 256. Then down the left-hand side, I start at 0 hours and then adding an hour each time because it takes an hour for them to double. And then we keep going until we get to 256 cells, which would be at 4 hours. So 4 hours is the final answer here. So let's try some quick questions on what we've covered so far to check our knowledge before we move on. So pause the video here and try these questions either by saying them out loud or writing them down. And when you're ready, play the video and we'll go through the answers. Okay, for the first question, it asked if a cell divides every hour, how many cells would be present after 5 hours? So you need to work this out in the same way that we did earlier. You have to lay out your times, so every hour, so 0 hours, 1, 2, 3, 4, 5, and then start with one cell and then just keep doubling until you get to the final answer, which would be 32. Second question asked, if a cell divides every 15 minutes, how many cells would there be after 2 hours? So again, we're starting at 0 minutes and we're basically going up in 15s until we get to 2 hours. Then we start with one cell and we just keep doubling until we get to the same line, and that would be 256. If a parent cell has 24 chromosomes, how many chromosomes will one daughter cell have? Well, this is actually quite a trick question. If the parent cell has 24 chromosomes, we know that each daughter cell needs to have the exact same number of chromosomes as the parent cell so that no genetic information is lost. So the answer to this one is still 24. Finally, it asks you to describe chromosome complement, what it means. Well, it's the characteristic number of chromosomes an organism has. So in humans, we have 46, whereas other organisms will have different numbers of chromosomes inside their nucleus normally. Okay, so basically what we're going to do is move on now and have a wee look at stem cells and specialisation. So we've just finished having a look at mitosis. So in this part, you'll be successful if you would be able to explain the purpose of stem cells in animals, describe the two types of stem cells and their importance, describe specialisation of cells, and finally describe the hierarchy within multicellular organisms. So firstly, what are stem cells? Well, stem cells are cells that are found in animals that are unspecialised, so they don't have a job or a function yet. These are specifically in animals. There is another type in plants, but you don't need to know what they are at National 5 level. So stem cells are unspecialised cells in animals that can do two main things. Firstly, they can divide in order to self-renew. This means they can divide to make more unspecialised cells. The second and most important thing about them is they have the potential to become many different cell types. So for example, in this diagram on the right, we have a stem cell in the middle, which is an unspecialised animal cell, which has the potential to become a liver cell, nerve cell, or blood cell, so different types of cell. The function of stem cells are that they're involved in growth and repair, which is really, really important. So they can create new cells to help with growth, or they can provide new cells to repair tissue that has been damaged. So if you get asked the function of stem cells, just growth or repair. This diagram just summarises what we've learned about stem cells so far. So stem cells are unspecialised cells in animals which can either divide to self-renew, so make more unspecialised cells, which you can see here, more stem cells, or they have the potential to become different types of cells. And their main function is that they're involved in growth and repair. The final thing you have to know about stem cells is where to find them or obtain them. So you can either take stem cells from the very early stages of an embryo, so the ball of cells that is formed when an egg and sperm meet, which we'll talk more about in KA1, or stem cells can be found in adult tissue in the body throughout life. So they're the two places that you can obtain stem cells from, so from the very early embryo or in adult tissues throughout life. So let's try some quick questions on what we've covered so far to check our knowledge before we move on. Once again, pause the video here, try the questions either by saying them out loud or writing them down, and when you're ready, play the video and we'll go through the answers. So the first question was, what are stem cells? Unspecialised cells in animals. What processes are stem cells involved in? Growth and repair. What two things can stem cells do? Well, they can divide in order to self-renew or make more stem cells, or they have the potential to become different types of cell. Finally, describe the two places stem cells can be obtained from the early embryo at a very early stage, or tissue stem cells can be found in the body throughout life. So the final part of this topic is about specialisation of cells. So we've just discussed unspecialised cells in animals, stem cells, but when they specialise, animal cells can form an amazing range of different cell types which all have different functions. Sperm cells, for example, are completely different in their shape and function to nerve cells. They are specialised to do their function or their job. Cell specialisation leads to the formation of lots of different types of cells, tissues, and organs. Cells are the most basic unit of living things, and if a group of cells work together to do the same function, then they can form a tissue, something like epithelium in the skin or muscular tissue in the muscles. Different tissues can then work together to make organs like the heart, lungs, and eyes. And finally, those organs can group and work together to form systems like the digestive system or the circulatory system. This layout here is called a hierarchy. So with the cells at the bottom, as they are the smallest unit and the least complex, and it works its way up through tissues, organs, and systems. Systems are the most complex here. So here's the hierarchy in a bit more detail. So the smallest and least complex at the bottom of this bit here, so cells moving up through tissues, which is a group of cells that perform the same function working together, to organs, which is a set of tissues working together, to systems, which are groups of organs working together, and finally the most complex is the overall organism, so like us, that is made up of systems, organs, tissues, and cells all working together. So multicellular organisms are organisms like us that are made up of many cell types, tissues, and organs. You can see some examples of the different cell types on the right-hand side. So red blood cells, sperm cells, neurons, and these ciliated cells that line the throat all look really different and are specialised to carry out different functions in the organs they're found in. This just shows some examples of systems and the organs that make them up in our bodies. You don't need to learn these. So one example is the reproductive system on the right-hand side, which we'll discuss soon in Unit 2. So the reproductive system is made up of organs like the testes and ovaries and other reproductive organs, which all have particular tissues and specialised cells within those systems that work together to do that job. The digestive system over here is another large system made up of organs like the stomach and large and small intestines, and each of these organs in the system are made up of tissues and cells that are specialised to their function as part of that system. Here's just one final example of a hierarchy to show how these all fit together. So the specialised cell in this example is a muscle cell which can contract and relax as part of its specialised function. Groups of muscle cells that perform a similar function group together to form a tissue, so that tissue here would be muscle tissue. Groups of tissues working together to perform a function form an organ, and in this case the heart is made up of different types of tissue, including that muscle tissue. And finally, the organ is part of a much larger system, so in this example the circulatory system, which joins with the rest of the systems in the body finally to make the organism. So cell, tissue, organ, system. You just need to remember that order. So let's try one final quick question on what we've covered so far to check our knowledge before we move on. So again, pause the video here, try this question, and then play the video and we'll go through the answer. So this question asks, which of the following shows the terms listed in the correct order of organisation in multicellular organisms? So is it organ, tissue, system, tissue, organ, system, or tissue, system, organ? Well, we know it's tissue, organ, then system. Cell would obviously come before this. You need to know the order of complexity, so it'd be tissue, organ, system. So B. So that's us finished learning about producing new cells. For this part, I hope that you can now explain the importance of stem cells in animals, the two types, and then their importance overall. Describe the specialisation of cells, describe the hierarchy within multicellular organisms as well. Please feel free to go back and watch parts of this video again in future if you need a refresher on this topic, but thank you once again for listening.